Good morning, everybody, and welcome to AnaEE's second TechUp webinar. This is the second webinar of our TechUp series, and we are happy to be joined by Klaus Koren from Aarhus University. So AnaEE is a European research infrastructure consortium with facilities all across Europe. I'm part of the Tech Centre, which is based in Copenhagen, and we have our central hub in France, and we have other centres dotted across Europe as well. I want to introduce our speaker today, Klaus Koren. He's going to be talking about chemical imaging in complex biological systems. He's going to highlight his and his research group's recent advances in this field and try and create an ambition to better understand these complex systems and untangle the connections between the biotic and the abiotic world. So, Klaus, if you'd like to begin.
Yep, sure. Then thank you all for coming and for the chance to talk here. I was not so much aware of this research infrastructure. I think it's very exciting. And maybe after the end of the presentation, you also think that optodes are exciting and that you can maybe use them in your research infrastructure in one way or the other. Yes, as said, it's going to be about chemical imaging in complex biological samples and how we can make chemistry visible. And as a disclaimer, this is me, I'm presenting, but it's the work of many of collaborator students and friends. And it's, science is a collaborative work. So I will try to highlight the people that actually do the work and not only the ones that present. Yes, but first a little bit about my background. I'm actually a chemist by training. So I studied chemistry in Austria and I did stuff like this, where you make dyes, molecules that are colorful and it's very nice. And then I got the chance to put those dyes in the biological sample. In this case, it was a cell line, and we could actually use the dyes are made to sense the intracellular oxygen. And it was a part of my PhD. And I was very excited about that because I could actually use something that I made in the lab to understand biology, to understand something complex. Yeah, and one way or the other, I ended up doing stuff like this. When you put a chemist on the Great Barrier Reef to sample corals, you can see that I look a bit alien to this place and totally not aware of what I'm doing. But it kind of tells the story that I come from chemistry going into new areas, going into different types of biology. And I think it's quite exciting because, as already mentioned, what I want to do is to understand how life and chemistry play together. So like how the biotic and abiotic world interact. And this can be different types of biotic systems like microbes, cells, plants, and so on. And those are interacting with terms of metabolism because certain chemical conditions determine what life is happening and not, and the other way around. And there's also environmental stressors, transport, signaling, and all those things are interfering and interacting in complex ways. And to understand that, I think we need tools, we need sensors to make those interactions visible, measurable. And ideally from small scale, like from a single cell, but also all the way up to the environment scale, from centimeters or meters or above. And ideally, we would like to see these chemical processes in real time, so extremely fast or in the time frame that is needed to understand the process. We have a bunch of different sensors we work with and develop. And here's just a partial list of those. And I will talk mainly about oxygen pH, a little bit about ammonia today, but there's a longer list and we work on optical sensors, which I will focus today, but we also have microsensors, which are very good tools, but I don't have the time today to go into them. But yeah, if you're interested, just reach out. And we look at different biological processes, mainly respiration type processes, photosynthesis, and also combine it with omics methods. Yes, but let's get started. How do we make an optode? How do we make chemistry visible? We need a dye, something like this. This is a phosphorescent dye, and then we mix it with a polymer and smear it on a glass or plastic support. It's very simple. That's what you do. And the interesting thing is this dye is reacting to an analyte. And here you can see the red dye. Can you see my mouse? Can I move the mouse? Yeah. This red dye that I just put on the film is very dim. Fluorescence or phosphorescence when there's oxygen around, and if we remove the oxygen, it gets brighter, and we can use this change in the intensity, this change in emission, to quantify oxygen. And similarly, we can make optodes, for example, for pH that would change their color from blue to yellowish when the pH changes from acidic to alkaline. So this is what we can do in terms of making chemistry visible. What we now need to do is to make this sensor come in contact with our sample. And here's what we can do. We can put this foil in a system. And here, example, we have some mud and a plant, freshwater plant, and then we put the sensor foil on top of it. And then we have the sensor in contact with the plant and with the mud. We can then put it in a little aquarium, have a camera pointing at it and some LED light source. And what we then can get is actually, first of all, if we have white light on, we can see the roots. We can see here the roots of this plant in the sediment. But if we now move to a blue light source and excite the probe, we can actually see oxygen. So we can see the release of oxygen around these roots. So we can see how the plant actually transports actively oxygen from the leaves to the roots to protect the roots from toxic H2S. Now we can see that in the day, when there's active photosynthesis, there's a lot of oxygen produced and there's a lot of oxygen transported to the roots, while at the night, when there's no oxygen production to the photosynthesis, we only rely on passive oxygen acquisition from the water phase, so there's less oxygen coming to the roots. And this really gives us a chemical image of what happens in this complex system, right? And then we can understand, well, what happens if we have oxygen depletion in the overlaying water? This will, of course, lead to less oxygen reaching the roots and eventually H2S intrusion and die-off of the plant. So we can really understand the chemical and biological processes by seeing and quantifying those. I'll give a few examples of what we can do. And the first example is on wildfires. It was a project that started by chance. It was a group in the US, in School of Mines and Colorado, that got interested in our technology and said, well, can we use that to study wildfire? And I'm like, yeah, sure, just come over and we have a look at it. And then they sent this guy, Alex Honeyman, and we played a little video. I'm not sure if you can hear the sound on your end. Yeah, because he wanted to study wildfire in Denmark. And if you don't know Denmark, Denmark is not the place to study wildfires. First of all, there's barely any forest. And second of all, it's quite wet. So we had to find our own way to make a little wildfire. And we made one in the parking lot where we took a soil core and, as you see, just burnt the grass on top of it. So we just created a little tiny wildfire. And we wanted to check what this wildfire does to the biology and the chemistry in the soil. And first of all, I thought, okay, well, when we burn it, it's going to be just, sterilizing the soil. There will be no microbes left and it will be just, quite boring. But actually, it turns out that soil is a quite good insulator. So the temperature gradient was actually quite steep and not so warm in the layers below. But what is interesting is when you look at the pH of the soil before and after the burn, it's a dramatic change because you have this alkaline ash layer that forms and then it will slowly percolate through the soil profile. And so after the fire, you actually see very deep down into the soil, see a very high pH. So it really changes the chemical microenvironment, which in turn will affect the microbes and the life within the soil. So we went on to study this in more detail. Here you can see Alex taking samples. This is the soil. We have one side for oxygen, one side for pH. So we look at the different oxygen pH concentrations, and then we take soil cores at known positions. And what we then can do is we can correlate where we took the sample with the chemical microenvironment. And then we can look at transcripts, and we can see, for example, certain genes. We see a nice correlation between for example, the oxygen concentration and some nitrogen cycling pathway, cycling genes, which is not surprising. But the interesting thing is just to see how complex soil is in this mosaic. If we just took layer by layer samples, we would not see any correlation because it's way more heterogeneous and you have this small microsites. So we can really use that to understand what's going on at the different positions, at the different spatially resolved. And the same for the pH environment. So we can correlate chemistry with biological processes. Here's another story that came up by chance. One of my colleagues has a friend who works with dung beetles. And then we were discussing and it was like, oh, can we study dung beetles with this methodology and what they do to soil? Like, yeah, sure, let's try. So we took a soil sandwich. So we squeezed a bit of soil between two glass plates. On one side of this little system, we put an oxygen optode, and the other side a pH optode. We put in some dung and some beetles, and then had a look what happened. So this is how it looks like. The dark part is anoxic. This is the dung that we put on here, and then you have the soil surface here. If I now play the video, you can see actually this is over 5 days, how the dung beetles take the dung and dig tunnels and bury this dung deep in the soil. So you can really see how the beetle restructures the chemistry, restructures the microenvironment in this soil sandwich. And the same is true for pH. If you look at pH, we can see a similar pattern where you can really see that the activity of the dung beetle really changes the pH deep down underneath the dung, so changing it to being more alkaline. And then you can see some images of that. And then just to be sure that it's really the dung that's moved and not something else, where we put fluorescent particles into the dung and made some fluorescent dung, because why not? And then you can actually see that the beetles actually move the dung deep into the soil. So it really moves the dung, and it correlates well with what we see in the chemical images. Yes, oxygen and pH are just our kind of bread and butter. So this is the stuff we do a lot, and it's the easiest in some way. But we also go beyond that, and one of the things we do is ammonia. So we developed an ammonia optode to look into the nitrogen cycle, and it was here's just how it works. It's published, so you can read it up. It's based on a pH indicator and a gas permeable membrane. And what's interesting is we can design it in a way that we can use a normal color camera to read it out, because there's one signal is changed by the ammonia concentration, while the other one is not. And it's a very large dynamic range, but not a super low limit of detection. But it's still very good for one part of a particular application, and this is agriculture, where we have a lot of ammonia, a lot of nitrogen added to the system. So really what we, of course, know from the planetary boundaries is, this is, I guess all of you know, that we are messing up with our planet, and one of the main things is that we put way too much phosphorus and nitrogen into the environment, and this is a problem. At the same time, we know that we need to give nitrogen to the plants to make them grow and produce food, so it's a bit of a dilemma. So what we want is that all the nitrogen we put goes into the plant and not elsewhere, right? So to understand that, we actually have to see what happens to the nitrogen in the soil. And the first test, what we did, this is Teresa Merl. She was a PhD and later postdoc in my lab. She really started this work and is a fantastic scientist. And what she did is she put some fertilizer granules or some fertilizer in the middle of the soil sample here, and then had a pH optode on one side and ammonia optode on the other side, and then we imaged the ammonia and pH over time after putting this fertilizer into the system. And this is how it looks. If we put just ammonium chloride in here, you can see that the ammonia diffuses out, and that you see this kind of donut-shaped pH ring around, and it will just grow and grow as the ammonia diffuses out. So we can track that in time and space. And if we now look at another type of fertilizer, like urea, if you put urea in, the picture looks very different. So it's spreading further out and the pH increases a lot. And this has to do, of course, with urea needs to be hydrolyzed by urease and so on. So you have a very different pattern of ammonia release in the system. And again, we can use our technology to track that and to see, okay, how fast do we actually see nitrogen disappearing? And then we can go in, and at those places where we have the circles here, we can take again samples of the soil destructively after our measurements, and then we can look at, for example, where's the nitrate, nitrite here for the ammonium chloride. You can see that most of the nitrate and nitrite is close to the where we deployed the fertilizer, while for the urea it's the opposite. So there's a lot of nitrate formed further away, which makes sense because it's two different types of diffusion. So we can really get an understanding how the nitrogen goes into the system from a local point source, like fertilizer. And also interesting and good to test, we tested with autoclaved soil, and there if we put urea in there, we don't see any ammonia release, because when we kill all the microbes, there's no urease. So we really can see that this is biological activity that makes the ammonia come out of the system. So now this is all fine and good, and we're happy that we can put our soil in the lab and put them in between glass plates. But this is, of course, not how it is in real life, right? In real life, we have soil out in the field, and things change, right? Here you can see a picture, and it was taken by Martin, who's a PhD student, just about to finish in a few weeks. And he did do some measurements in Germany, because this is not Denmark. There's no mountains in Denmark. And he deployed the system he will tell you about later here. And this is 2 days later, the landscape changed quite dramatically. So you can see that when you put things in the field, yeah, it's something different. And I guess you all are aware of that. So what we really need to do is we need to image what happens in real life conditions, right? We need to image chemical processes in there. And this is a bit tricky. And what people have done so far, and it's not been many, but a few have been attempting to do stuff like that, where they dig massive holes like ditches into the soil and then made windows, which is very nice. You can see the roots. And then you can equip those windows, for example, with optodes, and then you can do some chemical imaging. But of course, it's quite an endeavor to dig, several cubic meter hole and put a camera and everything in there. Alternatively, people some 20 years ago developed a system that actually was able to deploy an optode system at the deep sea, at the seafloor, where they designed it in a way like a inverted periscope that can be kind of stunk into the soft sediment. So that works fine, but it's only for very soft sediment. What we wanted to do is combine that and make a system that really can measure in situ without having to dig massive holes. And this is what MARTINIS is, our multi-analyte real-time in situ imaging system. It looks like this, and it's really the child of Martin, the PhD student who was working on that. And what's fantastic about this is that we put all the things needed for chemical imaging within a self-contained system. So everything is in this tube, and then we can code the outside of the tube with our sensors. It's all based on a Raspberry Pi, and it's placed in a 25 centimeter acrylic housing that is fully transparent. And the whole system is quite cheap. So I said Raspberry Pi, computer, camera, electronics, most of the things are 3D printed. And the camera can turn freely 360 and can move up and down. So we can really get a full view of the entire exterior. Can fully automate it, can image quite fast every few seconds, and still with a quite high spatial resolution. And the whole system is well below €1,000, so it's quite cheap compared to other scientific equipment. Yes, of course, we checked it can measure oxygen concentrations, also at different temperatures. And here you can see we just checked it. Just if you put it in the soil and keep it in one position, we can see the dynamics of oxygen depletion. But we can also make the camera move around. And then when it moves, it kind of moves in a way similar than when you take a panoramic image where you just scan and then the pictures are stitched together. It's the same it does here. So it has overlaying regions, makes image recognition, and then you can stitch the individual pixels together and get bigger images. So we can actually image, if we want to, almost a square meter of below ground soil chemistry. And the good thing is we can put all kinds of sensors on it. There's still some challenges if you want to take a system into the field. And I guess some of you have more experience with that than I do. Yeah, just how to deploy it best, how to make sure that we have a good contact from the soil to the optode, temperature compensation, long-term stability. And of course, we would like to do multi-analyte imaging. Yes, a deployment was the first challenge. And we bought a massive drill, and it kind of worked. But it's not that easy to make a big hole. And here you can see now the system in the soil with the camera, with the electronics out of the system, so you can see the tube. And here you can see the sensor foil pointing towards the soil. So we only covered a part of the total area of the tube. And then we put this the system in, and it only needs a power supply. The rest is fully self-contained. And we even went to extreme places. We actually went to Colorado and the Rocky Mountains. And sometimes if you don't have a lot of time to deploy, we actually needed to push the soil towards the sensor to get good contact. But what was exciting for us was that it was actually working. So we were able to have it standing in Germany in this field for 3.5 months and get continuous oxygen imaging of the below ground oxygenation. And we can actually see this heterogeneity, we can see hotspots, we can see roots, right? And then we can track it together with the precipitation data. For example, we can see what happens before a precipitation event, after, and after a heavy rainfall. You can see it gets totally water saturated and anoxic. So we can really, correlate it nicely, and we can get a full picture of what's happening. And this has been just published this year, and we're quite happy about it. And we also try to use it now, and this is a study that's currently in progress, and I hope it will come maybe out this year again or next year, where we combine this MARTINIS system with N2O emissions, with flux measurements, because we want to actually correlate what happens below the soil surface to what comes out of the soil surface. And here the data is also quite promising, and I think we can really show that getting this two-dimensional, highly resolved information of the oxygen helps us to better understand the dynamics and processes in the soil that lead to emissions. And then also, because we started this wildfire, we went back and we were actually invited to Colorado, and we put the whole system into the real wildfire. So this is not parking lot wildfire; it's a real big, massive wildfire. As you can see, a lot of area burned down. And here's Martin, and together with some of the other postdocs, deploying the system. And as you see, I'm trying my best to be helpful, even if it's only making shade. But it was a very interesting project, and as well, it was a bit challenging because of the rocky mountains with a lot of stones in the soil. But we managed. That actually shows that the system is quite capable of different systems. Yes. And as mentioned, one of the challenges is that we want to do multiple analyses. So measuring oxygen is nice and important, but it would be good if we can measure different types of analytes at the same point in time and space. Yeah. And this is a project that Andre, a postdoc in the lab, has been taking on. And the way we did this in collaboration with people from Southern Denmark University is to make a grid optode. So what we did is we hacked a 3D printer, as you can see here, and made the printer make some pattern. So we had spots, small spots for pH, oxygen is basically like a checkboard. We have a different, we can make all kinds of patterns that we would like to, and then we can know the spacing between the different pixels or the different dots. And then we can, this is the image of a sediment, this is the full image of the oxygen and pH dot. And then we can extract only those that are sensitive to oxygen and only those that are sensitive to pH. And then we do interpolation. So it's kind of like similar what your color camera does, because on your camera you also have red, green, and blue pixels, and then interpolates between them. So what we can do here, so we have some areas where we know something and some where we don't. So we then do interpolation, and then we get a full picture. Of course, with a less spatial resolution, but it's still a full picture. And here we can do the same, then at the same time measure oxygen and pH in the system. That we can get a very nice agreement with other reference methods. So it's a way, in a way, to get a very flexible platform where we can design which kind of parameters we want to measure next to each other. Yes. Yep, and now we'll jump to something totally different. And now it was all about soil and stuff like that. Now we'll talk about corals and aquatic systems. And I hope that's okay. I'm not sure what your backgrounds are, but yeah, corals, as you know, build this massive, beautiful reefs and are one of the ecosystems most threatened by climate change. So it's really an interesting topic. And when I did my postdoc in marine biology, I was taken on a field trip to the coral reef, which was very exciting. Though I didn't really know what I was supposed to do there because I was a chemist and had no idea. And I still don't know all the species names and so on. But then the idea was like, can we make the chemical microenvironments of corals better understandable, visual? And I was like, how can we do it? Corals are this, massive and have this 3D structure. But can we maybe spray paint a coral? Because I have nanoparticles, so I can make this sensor chemistry that I smear on a sensor foil. I can also turn them into small particles. The same chemistry, just in smaller particles. And I had this idea that we could spray paint a coral just with an airbrush and just nanoparticles. The biologists that were working with me were not that happy about it. We're like, nah, corals don't like to be taken out of the water, and then cold nanoparticles. No. But then, when you're on a small island, you can discuss it a bit longer, and maybe after some time, I convinced them, well, but we're spray painting ourselves, so why not try with corals? Anyway, at the end of the day, we decided, okay, we can take a small coral fragment and just try it out. So what we did is we took an airbrush, the ones you would use for painting model planes or cars, filled them with our nanoparticles, took a coral fragment, sprayed it, put it back in the aquarium, and then imaged it. But before we did this, we actually took a coral skeleton, so just the inorganic part, the calcium carbonate, and spray painted this just to see that the particles, how we can visualize them. And then we changed the oxygen in the water, and we can see that the ratio changes according to the oxygen concentration as expected. So we can actually get an image of oxygen. And this is now what happens when we take a real coral. So this is the coral fragment. This is how it looks under UV light, and this is how the oxygen on the coral fragment looks like. And it's quite exciting because you can actually see this is this three-dimensional shape, this structure that has so much information, and we can actually code it and get a picture of the oxygen, get a chemical visualization of what's happening. And I was quite excited and was quite happy, and we can actually see features like, for example, in the dark here, the algae will draw down the oxygen quite dramatically. You can see differences between the polyp mouth and the cenocyte. That was very nice. Turned out to be a nice little study, and actually ended up in a biology textbook, which is something I'm a little bit proud of because it was just a random idea. And so, but sometimes you have to try. But this is not the end of the story, because what we then did is people were like, yeah, cool that you can do this in stagnant conditions, but when a coral is in the water, there's flow, there's water coming in waves, and what happens to the oxygen exchange? Like, what happens to the oxygen that comes out of the coral or goes into the coral? And in order to do that, we had to develop a method called sense-PIV, sensor enhanced particle image velocimetry. And it's really something that was done in collaboration with Søren Ammerkamp, who is now at the IUW in Warnemünde. And here the idea is particle image velocimetry is a method where you put small particles into a system and then have a light sheet illumination of your sample and then a camera perpendicular to it. And then you track those particles in this light sheet very fast. And then if we can reconstruct the path of those particles, and by this you can reconstruct the flow field. So it's about tracking particles, and now we have particles already, and those particles are already fluorescent, actually oxygen sensitive. So we just combined that and try to make this method both able to track the flow, but also the oxygen. And this is how it looks like. I think it's easier to visualize. So here's a microsensor for reference. You can see those particles falling by. And then here you can, if you look carefully here at the border, at the surface of the coral, you can actually see that the particles moving a bit strangely. So they're moving a bit up and down. And this is because corals actually have cilia, like small hair at the surface that they can use to move the water. And what we could show with our method actually was that when you track those particles, you have the laminar flow on top, but then you have these vortexes, like little tornadoes going up and down in a repeating pattern on the surface of the coral. And those actually coincide with the oxygen. That is low oxygen, high oxygen, low oxygen, high oxygen. So you get a repeating pattern of oxygen and flow conditions. So this was quite intriguing. And actually the fact that we could now resolve that made us look a little bit further into that. And that basically worked from Cesar Pacheres, where he looked at what happens and why is this pattern emerging. And the interesting part is we see the highest oxygen on top of the mouth and the lowest in the surrounding tissues. And when you look at where actually the algae, the symbionts that produce the oxygen, then you see that they're actually not at the mouth, but at the surrounding. So what happens in our hypothesis is that you have a lot of chlorophyll, so you have a lot of oxygen production at light, and this oxygen can be toxic to the coral, to the animal. So the cilia are creating a vortex that will increase the mass transport of oxygen out of away from the coral, and then another vortex will transport the oxygen back down into the polyp mouth, where the cilia, where the coral actually needs oxygen for respiration. That's kind of like cycling the oxygen via the water phase. At least this is a theory that we came up with, and that we're now currently testing also with more different coral species and so on. So been back to the reef and looked at that in detail. So it's very interesting because we can correlate, again, biological function to chemical conditions and learn about how they interact with each other. So that's something I'm very excited about. And along the way, and if I say we, it's mainly Søren, realized that there's a method that we could use to make our time resolution better. And I will not go into detail with that, but we actually use the fact that the camera has frames and an interframe time. And if we correlate that with our pulses of our laser, we can actually do it in a way that we have one frame where we only see the excitation, but not the emission curve of the dye. So we get a bit dim image, and if the other one, we get a brighter image because we collect both the excitation and the emission. And when we take the ratio of those two images, then they also correlate to the oxygen concentration. And the interesting part here is mainly that this is a very fast method. We can actually measure extremely fast, so at a rate of 10 hertz or more. So we can have images every 10 milliseconds, so following extremely fast processes if you want to. And I just brought one example. Here you see a cuvette filled with nanoparticles, and then we're throwing in sodium dithionide, which is a chemical that reacts with oxygen and consumes oxygen. And if you look at that in the oxygen image, you can actually see those dithionide crystals falling down and depleting the oxygen around them. So you can really follow the chemical reaction. And this is recorded at 100 frames per second. So it's actually slowed down from real speed. So we have really, really fast dynamics, and we can actually follow a chemical reaction, more or less in real time. So that's very nice. And this method, I think, gives us more opportunities now to also look at very fast biological processes, chemical reactions, all kinds of methodologies. We're trying to improve that also for other analytes other than oxygen. Yes, and I think this was already it, bringing me to my favorite and final quote, which is, if the only tool you have is a hammer, you tend to see every problem as a nail, from Abraham Maslow. Although he's a psychologist, or was a psychologist, I think this is quite a good quote because it shows us that the tools we have available determine what we can do, right? And I think, yes, most of you will agree that having more tools available is helpful because we can attack new problems and not always do the same. So that I like to develop tools and also use them. And I recently extended this quote by, if the shoe fits, walk it until the high heel breaks, from Taylor Swift. And this is just to show, if you have a tool, it's also important to make use of it, not only to make new tools after new tools that nobody needs. So if you have a good tool, there's so many things we can use them for and learn a lot. So some of our oxygen sensors, we don't have to reinvent all the time because they're just working very well, and we can use them to study different systems. And I think that's what we also want to do, spread this technology, make people aware of it, and make them capable of using it to answer the questions they have and the problems they face, if the tools can be helpful. Yes, that I would like to thank those generous foundations that give money to some of the stuff I do, and otherwise wouldn't be possible. Big thanks also to my collaboration partners. I've been very lucky over the years to work with very nice people, and that's very important. Nice department here. And we're always open for collaborations, and if we're not having any secrets, we're always happy to share. Yes. And as a big thanks to my group. This is already an old picture, so by now some of them have left, but I've been very, very lucky to work with extremely smart and nice people over the last years that made all the things possible. So I was just, I'm just giving the presentations, but those people really did the work and developed lots of the things. And so there's more cool stuff to come. And if you're interested, just follow us or follow me on LinkedIn. Try to post some of our updates on recent science. Sometimes I complain about politics and share pictures of my dog, but that's about it. So thank you for your time and attention, and I hope it was worth the time. And yeah, I'm open for questions.
Well, thank you very much. That was a really, really interesting presentation. And I like that you covered several topics within your research scope and looked at lots of different technologies. So I think that's going to be really interesting for the Technology Centre and other people who are present right now. But yeah, I'll leave the floor open if anyone's got any questions. I can see, Ivan, you have a question.
Klaus, very exciting talk, especially the wildfire in the parking lot. I will not forget that. It's good to hear that there is a sensor lab in Aarhus. I think I heard it before, but I was lost in my mind.
Yeah, I took over the lab more or less from Nils Peter Revsbech, who was doing pioneering the microsensor work, and we still do a lot of microsensor work, but now also do the optics. Yes.
I was wondering whether an optode could be fitted to an ecotron. I'm based in Antwerp University. I'm running an ecotron and also an open air facility. So an ecotron, I'm not sure if you're familiar with the concept. It's basically a lysimeter with the climate control on top. So we put sensors in the lysimeter from the site, soil water content and others. So I wonder if a window could be fitted on which you could mount the optode equipment.
Yes, depends on. Would have to see the system, right? But generally, the nice thing with the optical sensors is, as long as you have a transparent window or something where you can see through, you can more or less access them remotely, right? So you don't have to open it. So compared to normal sensors where you have to have a cable to get the data, we get the data via light. So if light can penetrate, we can more or less get there. So
how big would the window need to be? Just to have an idea, because we have lots of sensors in these units. Some ecotrons have very large units up to multiple cubic meters. Ours is 1 cubic meter. So is it something like a few centimeters, or should it be bigger, like 10, 20 centimeters? Because that
depends, but we can make our sensors from small spots of a few millimeters in diameter to tens of centimeters square. So it's very much, we're very flexible in that. And it basically depends. You can make if you make a small spot, you of course get a point measurement. But if you get a bigger area, you can do this imaging that I showed and get a more two-dimensional representation. So this is quite flexible. So we've done it.
It seems to be possible, and if the windows are mounted on multiple ecotron units, then you would, in principle, could mount several optodes, since they're not expensive, apparently. Or you could go around and mount it successively in kind of rotation.
Yeah.
I think it opens possibilities.
Yes, definitely. Are you in Hasselt, or where are you?
I'm in Antwerp. In Hasselt, there is another ecotron. That's a macroscale ecotron, which has bigger soil chunks. We have smaller ones, and there's another one. That's an indoor ecotron in Jean Blou in the south of Belgium. So there's a high concentration there. So I'll tell my colleagues.
Because a former postdoc of mine, Pauline Rummel, she's going to Hasselt now, and to also help them with sensor integration, I think. So if you want, you can also. She will be close by. I think she's starting next month.
Okay, super.
Yes.
I have more questions, Rachel, but there's more people raising hands, so they go first, I suppose.
Okay, we'll go to Janko and then we'll come back to you.
Hello, everyone. Thank you for the presentation. For me, it was like something completely new, out of the blue. It's really novel, and I learned a lot of stuff today. Actually, I have a few questions, but I will emphasize one. So I'm a forester. I have background in forestry, and I'm interested in forest and all kind of stuff related with forest. So I'm wondering, could this like a technology, these techniques, optodes help us to understand the soil microenvironment? So to be like a direct, can these devices help us like to see where, for instance, truffles forms in the forest?
Could be. I'm actually. I have a little sweet spot for fungi, so I would actually like to use that also to study fungi. And I have this idea, this witch rings when this in the soil, when the grass is growing higher and rings shapes. So I would like to actually place them at this forefront and see how the fungi and so on grow into the sensor, right? So I think it could be possible that you deploy a system and then see how the microbiome and how the fungi and so on grow into it. And I think, for example, this tube, we could design it in a way that we have one slice where we measure oxygen, then have a free space, and then measure pH and so on. So we can see also the structure and the chemistry side by side in a way. So yeah, principle, yes, depends a bit on the system, but it would at least be fun.
And I can also like just agree with your like a quote about this, because today how I understood that science is driven by the devices, but maybe it's not so good. But I have also a question related with the urban forestry, and how you see that optode like technology can be applied to understand to understand the processes like in urban greenery, in urban forestry, because they have let's say a limited space for the root and to monitor root activity or soil oxygen levels.
Okay. It's something I've not thought about at all. So that would be very new for me. I'm a background in chemistry, so all this no, applications are all new to me. But if you have a certain question, I think one could think about integrating them in if you have a growing, I don't know, trees growing in a very defined space. You could put them in and see how the roots change and so on. So it could become like a rhizo camera, like an extended rhizo camera system, where you not only get the root structure but also chemistry around. So that is something we're considering. We're actually considering if we should go commercial with this system, but I'm not sure. But yeah, would be interested to see if there's a demand for it, because I think it could be interesting to get this technology out and make it accessible for people to then, apply it in whatever they are interested.
Yeah, to test and valid. Yeah. Okay.
Yeah.
Thank you so much. Thank you so much for the presentation. It
Yeah,
was
thanks.
very nice.
Always nice if people are interested.
Cool. Ivan, did you have some more questions?
Oh yeah, if there's still time.
Yeah, of course. Yeah.
You showed that combined measurements of ions are more, well, no, it was oxygen, I think, and pH. They're a bit more challenging, but it can also be done. So what about combined measurements for different ions, like nitrate, ammonium, and phosphate, typically the things that a plant ecologist, biogeochemist would be interested in?
Definitely, and that's also the next frontier we're going into. So there's some optical sensors for things like ammonium and calcium and so on. So those are available, but the problem is that they are normally pH cross-sensitive, so they would also respond to changes in the pH. But now with our patterning approach, we can actually, and that's I think quite elegant, we can make a spot for ammonium and next to it a spot for pH, and then we can actually correct for the cross-sensitivity in very, very close proximity. But this is really I hope that we can get this in the next few months. So that's my postdoc, Andre, is going to work further on that. And I think this is quite promising. And I fully agree that this is where we should go. And that's, really tapping into those nutrient cycles and ions. So that would be...
The instruments always delivers concentration, right? Or a variable related to concentration.
Yeah, it measures some kind of emission, some kind of optical change, and then we have to calibrate it to concentration or activity, depending on.
Yeah. Then I'm always stuck with the conundrum that, for example, a low concentration of an ion can indicate low mineralization, but also a high plant uptake. Can these optodes sort of help disentangling that in some way, or do you need to combine other types of measurements with the optode?
I think what we could do, of course, we could look at the time series, right? We can look at dynamics and see how roots will take them up, right? Have roots growing into a patch of nutrients or ions, and then see the dynamics of how the nutrients are depleted, right? In that sense, and then that's one of the upsides of the optodes is they're non-invasive. Well, they're invasive in some extent, but they can stay there for a long time, and you don't have to go and take samples out to analyze them, because you can just follow that over longer times, right? So we can follow over multiple days and then see the dynamics. So I think this could be a way to look into those processes. But I'm happy to discuss more. I said I'm not a expert in soil processes.
Okay, great. Yeah, thanks.
Yeah. And of course, combining with isotope methods and so on. Yeah, of course. It's always nice.
Yeah. Well, what we use is resin caps sometimes. They accumulate ions, of course, and then you get many ions, but you don't have anything temporal and you don't have anything spatial, and you're assuming it behaves like plant roots. So it's maybe it can be a complement. Maybe you also have to insert it in your system and take it out. So the flexibility here is much bigger in terms of spatial variations that you can see, and temporal tracking, and the non-invasiveness, etc. So
but they're very
complement, I think.
Yeah, but they're very nicely complement. We worked also with these DGT gels, diffusive gradient thin films, that take up multiple ions, and then you can scan laser ablation and so on. So we also worked a little bit with those, and they're quite powerful. But of course, they are just they accumulate, so they are kind of a dosimeter that will over time, and
Okay.
we get limited temporal resolution. Yeah.
Okay, thanks.
I can see Adriana has a question. So if you want to ask.
Yes,
I
of
think-
course.
okay.
Good morning. Yes, can you hear me?
Yes.
Yes, yeah, of course. Very interesting presentation. Yes, I'm not a real expert in the field, but so I have just a curiosity regarding the experiment for the wildfire in the small box.
Yeah.
I think there are also influences at greater depths as well. I see also you make some field measurements, but is it possible maybe to also model what happens in the lower layers somehow?
Yeah, definitely. I mean, yeah. This was, as I said, this was just a a little fun experiment that we did, and just to show that we can actually see something. And I fully agree. Of course, this is a very model system, but and I think that's actually one of the more exciting things is that we see that this ash and this pH change goes deeper, right? And we could also, would be super nice to model this together with hydrology and see how does it percolate and what does it do. Also, the wildfire changes the hydrophobicity of the soil surface and so on. And so I think there's many aspects that can go into that, and I think that would be lovely to go deeper. Yeah. So yeah, it's just to show that we can do that, and then I think it would be yeah, we would also like to expand it also to, for example, other metals, like I don't know, like heavy metals that could be produced from wildfires. How do they actually persist in the soil, right? Will they get washed downwards, and how do they accumulate or not? So I think that there's many aspects I would like to go into, but I was not able to get more funding for this.
Yeah. Okay. Yeah. Anyway, congratulations. Very interesting. Yeah.
Thanks.
I think that the technology you're working on right now is definitely something we can try and find a way of integrating with our project, or several of our projects here with AnaEE.
Sure. Yes,
It would be
please.
nice, and we'll be in touch.
Cool. And thanks for the invite. It's very nice, and I will try to keep myself updated on your activities. It's nice.
Yeah, no problem.
Yeah.
All right. Have a wonderful day, everybody.
Have a good
Bye.
day. Bye-bye.
Bye. Bye.
Bye.